warm welcome to everyone. We would like to wish everyone a good morning, a good afternoon, or a good evening, depending on where you are in the world. My name is Samantha from Safeopedia, and here at Safeopedia, it is our mission to support the EHS professionals, operational folks, and any safety-minded individuals with free safety information, tools, and education. I'd like to extend a huge thank you to the dedicated professionals for the great work they do on a daily basis. Registration is now open for our virtual safety conference and expo, Safety Connect. We have 3 days, October 19th to 21st, packed with great speakers, exhibitor booths, hundreds of door prizes, 3 grand prizes, and 1.7 continuing education units, all for free. Where else can you get this? Seriously, 1.7 CEUs for free. Visit industryconnectsafety.com for more information and to register for free. Just a reminder, this webinar is being recorded and we will send out a copy of the recording as well as the slide deck to everyone in a few days. Let's keep it interactive. So make sure you get your questions into the GoToWebinar console and we'll get to them at the end of the presentation. Today, we're proud to present chemical, pharma, and laboratory gas detection issues and answers. This Safeopedia webinar has been made possible today by GFG, a preferred manufacturer member of 80 Safety Network. 80, together we win. It is now my pleasure to welcome back today's presenter, Bob Henderson. Bob is the president of GFG Instrumentation, Inc., a leading supplier of portable and fixed gas detection and indoor air quality monitoring products. He has over 38 years of experience in design, marketing, and manufacturing for gas detection instruments. He was one of the original team who helped to start Biosystems back in 1982, where he was director of marketing. Later, he went on to become vice president of marketing at RAE Systems, and later yet, vice president of business development at BW Technologies. For the last 14 years, he has been the president of GFG Instrumentation, Inc. in Ann Arbor, Michigan. I'm very excited to have you all sit back and let Bob talk your ear off. With that, Bob, please take it away.
Thank you, Samantha. Well, yeah, it's a lot easier for me to keep going for several hours than to cut it down to one meager hour, but I'm going to do my best. Chemical, pharma, laboratory gas detection issues and answers. That's a pretty spacious topic. It covers a lot of ground, a lot of issues. So we'll get right into it. So if you're not familiar with GFG, we've been in business since 1961. The only thing that we do is gas detection. We are a worldwide manufacturer of fixed and portable gas detection solutions. So don't forget my email address and the contact information at the end of the seminar, because we are here to help you. We have a passionate interest in the safety of our customers and gas detection issues, and providing the best advice that we possibly can with regards to how to solve your often pretty tough gas detection needs and issues. Anyway, when it comes to chemical, pharma, laboratory applications, typically the sites share some similarities, but of course, these different businesses can be vastly different from each other as well. So safety, hygiene, facilities, production managers deal with an extremely wide range of atmospheric hazards monitoring applications and activities. Managers need to anticipate the critical requirements ahead of time. So the idea for today is to help you identify some of these issues and considerations. Better to think about them up front than to deal with things after the fact. At the end of the day, it is critically important that your gas detection equipment must be fit for purpose. So the more you know, the more likely it is that gas detection decisions that you make will lead to equipment that is fit for purpose. So if you're sitting down for the first time, and probably this is not the case for most of you, for some of you it might be, and for some of you we have, looking at the audience makeup, we have lots of people from the distributor side of the divide, we have contractors, we have site managers and safety personnel. So we've really got a diverse group of people who are signed on for the webinar. But whatever your role, the more detailed grasp you have of the activities and risks that involve atmospheric hazards at your site, the better. And you really do need to drill down to make sure you understand what is most important. If you've been hearing about some issues on a regular basis from your fellow employees, well, that's a good starting point. But remember, there might be something else of equal importance or several other something else's of equal importance that you won't know about unless you really drill down and make sure that you've taken a look. But part of this drill down process is to make sure that you are currently meeting all monitoring and safety and regulatory requirements. You need to figure out where you need to make improvements. And remember that gas detection issues are not necessarily and solely limited to safety. Toxic exposure limits are getting lower every year. There are a lot of facilities protection issues that overlap with gas detection requirements. So there's more to life than only safety. Chemical and pharma industry managers are involved with a wide range of safety and hygiene gas detection. You have personal exposure monitoring, confined space, hot work, toxic materials, vapors, gases, hazmat, emergency response, other activity-based monitoring. That's a really wide range of activities. Now, we uh, last the webinar that we gave most recently was a deep dive into the oil and petrochemical industry. So you can always have another look at that earlier webinar. It's posted on Safeopedia. It hasn't gone anywhere. So if you're having some curiosity and would like a little more detail on the petrochemical side of the chemical industry, have a look at that earlier webinar. But petrochemical plants typically are converting feedstock natural gas or feedstock crude oil or partially processed oil or bitumen into new products that are more saleable. Um, on the chemical plant side of the refinery, we're talking about the production of olefins. These are hydrocarbons that have a double bond or are unsaturated. That would typically include VOCs like propylene or ethylene or toluene or cumene or xylene, a whole lot of chemicals that end in ENE. You also have the production of aromatic hydrocarbons and chemicals. These are chemicals that have a ring structure. Um, and you have a multitude of other VOC, solvent, and hydrocarbon products that are being produced. Other chemical plants specialize in the production of inorganic chemicals. Some of the leading inorganic chemical categories would be plants that specialize in the production of sulfur, sulfide, sulfuric acid, chlorine, ammonia, hydrochloric acid, chlor-alkali plants that produce lye, caustic soda, potash, plants that produce phosphorus and phosphates that are used in fertilizers. The hazards that you're going to be concerned with are a direct consequence of the processes and the materials that you're dealing with at that particular plant. The chemical industry gas detection requirements can, again, include everything from personal protection to production, process, facilities protection, industrial hygiene, community, such as fence line, EPA, nuisance odor, disaster response. You better have thought through what you're going to do in the event of a spill or an explosion or a fire. You have construction shutdowns where you are deliberately doing as much maintenance as you can at a time when the plant is partially or completely shut down to facilitate the shutdown maintenance and safety procedures. And of course, confined space. There's more to monitoring than only confined space, but confined space is a big deal. You have routine entries that are going on all the time. You have large scale entries that can be ongoing for weeks or longer at a time during lengthy shutdown periods. You have entries into inerted vessels. You have confined space entry that involves hot work, and you have all sorts of special confined space entry procedures like catalyst rebuilding, where you're deliberately entering an inerted confined space to repack and rebuild the catalyst that you're using to make your products. And this is a daunting set of gas detection tasks. Watch out at chemical plants for the presence of hydrogen. Hydrogen can be easily measured if you want to measure hydrogen, but it also causes problems with the proper performance of some of your other sensors and may have an effect on the proper performance of your 4-gas or 5-gas instruments. So petrochemical plants produce olefins and aromatics by catalytic cracking of petroleum fractions. Cracking converts high molecular big molecules into more useful, more saleable, lower molecular weight molecules. Catalytic cracking uses and produces hydrogen. Chemical plants that produce ammonia by using natural gas produce hydrogen and then use nitrogen plus the hydrogen to produce the NH3. Gas concerns from the hydrogen standpoint, well, be careful what kind of combustible sensor that you're using. There are about 4 different popular types of combustible gas sensors out there. The oldest type that has been around for a long time uses catalytic oxidation. It detects the hydrogen and any other combustible gas by burning it. An increasingly popular kind of combustible sensor now is using infrared absorbance to detect the combustible gas. That's a great way to detect methane, but infrared sensors cannot detect hydrogen. Hydrogen does not absorb infrared radiation. It does not absorb. Molecules do not absorb infrared light. So they are IR invisible, which means you need to use a different method to detect the hydrogen. Hydrogen is also an interferent of carbon monoxide sensors. Now, a standard carbon monoxide sensor or a dual channel COH2S sensor can sometimes have a cross sensitivity to hydrogen of up to 40%. So if you have 100 parts per million of hydrogen around, the CO sensor is going to show a reading of 40 parts per million. So it's responding to hydrogen, not responding to the carbon monoxide. If you have a hydrogen nulled or a low hydrogen sensitivity COH2S sensor or single channel CO sensor, it depends on the design, but in a dual channel hydrogen low hydrogen interference 2-channel dual channel COH2S sensor, probably you're going to see a relative response of 20%. With a single channel CO sensor that has been deliberately manufactured to really reduce the cross sensitivity, it might only be 5%. It's not going to be zero. So when a manufacturer uses the term hydrogen nulled CO sensor, it's not nulled all the way down to zero. About as good as it's going to get is a 5% relative response. So that means that if you have, say, 1% LEL hydrogen, which would be a concentration of 400 parts per million, you're going to get a reading of 5% of 400. So, that's quite a bit of hydrogen. That's going to look like you have on the CO channel. You're going to be in alarm for carbon monoxide if you're setting it at the permissible exposure limit. So even though it is hydrogen nulled, it doesn't stop you from having alarms that might be due to the presence of hydrogen. If you need to measure the hydrogen, you can directly measure it by using an electrochemical sensor that's substance specific and designed to directly measure hydrogen. If you are using an infrared LEL sensor to measure combustible gas, I would strongly urge you to include a hydrogen sensor in the same multi-sensor instrument. If you don't do that, at least get familiar and comfortable with the effects of the hydrogen on your carbon monoxide sensor, the one that you're using in that instrument. What are the most common chemical industry atmospheric hazards? Well, I can just list the ones that we run into the most frequently, but the complete list is extremely lengthy. Right at the top of the list, you have oxygen deficiency and oxygen enrichment. You have the presence of toxic gases in petrochemical plants. Right at the top of that list, you have H2S. You also have carbon monoxide. You have the presence of combustible gases. And typically, you're using a single gas H2S or a 4-gas or 5-gas detector with LEL, oxygen, carbon monoxide, H2S, and increasingly PID sensors for the measurement of volatile organic chemical at the toxic exposure limit. So some of the other commonly encountered site-specific chemical plant hazards include chlorine, ammonia, sulfur dioxide, nitrogen dioxide, benzene, CO2, HF, hydrofluoric acid, ozone, cyanide, phosphine, specific sulfides like methyl mercaptan that are used to synthesize other chemicals such as fumigants and insecticides and all kinds of poisonous stuff. The list is really lengthy. And fortunately, you have direct reading sensors that you can plug into a multi-sensor instrument that are capable of measuring just about anything that you would like to measure. With pharmaceutical industry gas detection concerns, it's a different list, and it comes from the type of processes and activities that are done at pharma plants. So what does a pharma plant exist to do? It exists to do industrial scale synthesis of pharmaceutical drugs. That's something a little bit different than an R&D laboratory or a pilot production laboratory where you're working out the kinks. But once you're talking about a full-sized pharma plant, then now the emphasis is on industrial scale production of the pharmaceutical drug. The processes that are going on include extraction, which can use and does use solvents and gases, culturing and fermentation, where you're actually growing the product or the precursor product, which will then undergo further processing. You're using a lot of the same techniques that you use in a bakery or a distillery to produce ethanol or to produce bread from yeast. So you have a lot of the same concerns. You have milling, granulation, coating, tablet pressing. You have to turn the active materials into a finished product that, besides the active ingredients, includes inactive excipient ingredients that are just there to give you something to swallow, along with the stuff that is really going to have the pharmaceutical effect when it hits your system. And you have packaging. Each of these stages and steps is associated with different kinds of atmospheric hazards and gas detection considerations. The pharmaceutical plants are sometimes the size of small cities. The plant, which is in the inset image at the top of the PowerPoint slide, happens to be the Pfizer pharmaceutical plant in Kalamazoo, Michigan, not too far down the road from GFG. And that gives you some idea of the size and scope and amazing complexity of this industry. At chemical plants, a lot of times the most important or large-scale major confined space activities occur during planned shutdowns and planned maintenance procedures and periods. At pharma plants, CS entry often occurs after every batch as part of procedures to sanitize and prepare the equipment for the next batch. Pharma plants have a lot of confined spaces: mixing tanks, manholes, fermenters, tunnels, bins, pipelines, pits, equipment housings, autoclaves. What is an autoclave? Well, an autoclave is a large oven where you are curing things. It might be at high temperature, it might be at under ambient temperature and pressure conditions where you're flowing a fumigant gas like ethylene oxide into the autoclave to disinfect and sanitize plastic materials that you can't sanitize by putting through a high temperature sanitization process. You have water and wastewater systems that are, again, the size of the systems in a small town. You have sewers, you have all kinds of enclosed areas and pits. It's interesting in this picture in the lower left-hand corner, if you look kind of carefully, you'll see the fermentation vessels, and you'll also see that the vessels are built down into pits and overflow reservoirs that will keep any leaks or catastrophic failures in the plumbing, will keep that material from going everywhere. And you'll notice that there are stairs that go down to the pits below the level of the bottom of the tank. So the tanks themselves, of course, are confined spaces. The pits are confined spaces. You got confined spaces in every which direction. One of the really different things about life in the big pharmaceutical industry is the complexity of the manufacture of the products, which is also distributed across multiple sites. So you're doing different steps at different locations. And by the time it all comes together, it's just darn near miraculous. This is not a political talk. All that I want to do today is to point out how amazing it is, all of the steps in production that go into making a vaccine, for instance. So how do you make a vaccine? Well, everybody, every evening, whether you want to or not, are hearing news items about vaccines. So what goes into making a vaccine? Well, Pfizer has one of the, not the only, but one of the leading um, vaccines that have been approved now by the CDC. So different steps. The first stage is DNA plasmids that code for the spike protein in the COVID virus particle. What is a plasmid? It's a loop of DNA. It's not a virus. It's not alive. It's just a little loop of DNA material that is capable of being replicated. It's infused into E. coli bacteria, and the bacteria are cultured. So you're growing them in massive quantities. You harvest the bacteria, you rupture the bacterial cells, you remove and purify the extract to recover the DNA product, and it's on to the next step at a different facility where the DNA is used as a template to build something called messenger RNA strands. Messenger RNAs are kind of the mirror image of the DNA strands. They're used as the template to make additional strands of DNA. So this whole process starts with relatively very few strands to a logarithmically higher amount of DNA that is produced through this amplification process. So in the second stage, the mRNA is purified, frozen in plastic bags, transported to yet another site. Each of these bags can hold up to 10 million doses. It's a lot of vaccine in one little bag. The third stage now is where the mRNA is combined with lipid nanoparticles to form an injectable suspension. Um, a lot of times when I hear the word nanoparticle, I'm thinking about science fiction movies like the one with Keanu Reeves, where killer nanoparticles are dissolving people and the world and animals and everything else. That's not what we're talking about here. What we're talking about is a suspension of lipid, which are naturally occurring fats that you have present in your cells, that are produced in a very size specific range. So you have a very uniform distribution of particle sizes, and you use that in the injectable suspension. And then from there, you still need to package the vaccine in vials, box it up, put it into disposable syringes, and transport it to medical centers. So what's going on at various stages? Well, in the culturing stage, one of the dominant things that you're worried about is the production of carbon dioxide. You might have the production by fermentation of alcohol, although that's a little bit less likely. Of course, you're using alcohol copiously when it comes to sanitization procedures. And you have oxygen issues, you have liquid nitrogen issues. In the packaging phase, you often are talking about materials that need to be disinfected. Well, you can't just put the vaccine into a high temperature and boil it to sanitize it. That would destroy the DNA and the molecules that you need to be in perfect shape when it goes into somebody's arm. So On the packaging on the outside, often you're sanitizing it by means of something like ethylene oxide. Uh, Ethylene oxide doesn't do any harm to the packaging or the plastic. When it's in the van, it has to be kept at a extremely cold -50-degree temperatures, and generally that's being done with dry ice. So once again, you have carbon dioxide concerns. It's just an amazing process, which has steps that are occurring often in entirely different states. And then you finally get to the assembled product at the medical center where people are lined up to get their shots. It's not an easy process to administer, and each step has its own gas detection concerns. Especially common atmospheric hazards, again, oxygen deficiency, especially when it's due to displacement by nitrogen at not necessarily in the process that we were just talking about, but often in a medical lab, in a hospital, in a pharma plant, you will find samples that are being frozen for later use in extremely low temperatures in a bath of liquid nitrogen. And what you're looking at here is essentially a giant thermos bottle, or technically what is called a dewar. Another name for a thermos bottle is a dewar flask. Same deal, only large. So you have a lagoon of liquid nitrogen on the inside of the dewar and a heavy swing down lid that you lift when you need to put something into the dewar or remove something from the dewar. When you swing the lid up, you'll see a boiling cloud of white vapor. That's not nitrogen that you're looking at. That's actually frozen moisture in the air that comes in contact with the very cold nitrogen. But it shows you what happens to the nitrogen. It's cold and it settles to the floor. So if you're in a closed and enclosed room, you are dependent on having really good ventilation that is continuously in place, turned on, and is sufficient to uh, ventilate that enclosed room, or cryo bank, as we call these areas, with fresh air. If you have a loss of power or a loss of ventilation, or your ventilation just for some reason becomes ineffective, or if you have a break in the plumbing that is introducing the hydrogen into the dewar, or the piping that is taking either pressurized hydrogen or liquid hydrogen through the facility, all of those sources of leakage can result in an oxygen displacement. So you have a very high obligation to do something to measure the oxygen concentration in the area and tie it into alarms, and often with meters and alarm lights and high intensity horns on the outside of the room, so you know that something is wrong before you open the door, not after you open the door, go in and lose consciousness. Carbon dioxide, again, it's produced by fermentation, it's produced by bacterial culturing. It's also part of um, it's what dry ice is frozen carbon dioxide. So you have lots of places where you should be measuring carbon dioxide, and in many cases you want to include a carbon dioxide sensor in your multi-sensor instrument. So you're looking at CO2 at the same time you're looking at combustible gas and the rest of the hazards. Alcohols are pervasively common in pharmaceutical laboratories, in pharmaceutical production areas. It can be a byproduct of fermentation. It can also uh, be part of sanitization procedures. We'll be talking about combustible sensors and the effects of some of these gases on LEL sensors in a little bit. But if you're using traditional catalytic LEL sensors that detect combustible gas by burning the gas on a bead, an active bead on the inside of the sensor, that bead is actually made out of alumina. It's alumina that is treated or doped with a catalytic material that allows oxidation to occur, but the bead itself is made out of alumina. So methanol has a high corrosive affinity for alumina. And methanol is notoriously bad at, or good at burning out catalytic type LEL sensors. So if you are using a lot of methanol or other alcohols as well at that site, then either be prepared for replacing that catalytic LEL sensor more frequently, or you might want to switch to a different kind of sensor like an infrared sensor. Ammonia. Um, you use a lot of ammonia in refrigeration systems that run through pharmaceutical plants. You don't generally run your refrigeration systems off of the liquid nitrogen. It's just a little too dangerous. You use ammonia to cool down what is called brine, a liquid that is then pumped in pipes around the site or wherever you want the cooling to occur. And it's the brine that transfers the cold temperature to the ambient atmosphere. You recirculate the brine back to the countercurrent ammonia refrigeration system and compressor, and that's what's going on. So you have ammonia near the compressor, you have ammonia reservoirs on the ceiling, you have the potential for the release of ammonia that can get into vent lines and into plenums and all of these places. You probably should, if you've not already, think about adding ammonia monitoring. Sometimes the best approach is fixed, sometimes the best is portable. Often you need a combination of fixed and portable detection to be safe from all standpoints. Laboratories come in all sizes and level of sophistication. I won't tell you the name of the company, but I will tell you that the three inset pictures in this slide um, were taken in an area where we were adding some oxygen monitoring, and this was a prominently named medical slash pharmaceutical company. This was a building that had over 100 laboratories in one large complex. Each of the little labs was very small. And bear in mind, the researchers that are doing their job in these little labs are probably God only knows where they're too smart for Mensa. Their IQ is so high that it would shame the other Mensa members to have these guys in the same room. They are multi-million-dollar a year PhD medical researchers, and yet they are not focused on safety, they are focused on their research. So you'll notice this cluster of nitrogen and combustible gas cylinders and dewars. Uh, no visible at this time monitoring equipment. You see these orange colored cases off on the back wall of this little lab, a different little room. These are running under a nitrogen purge. So on the inside of these little cabinets, you have zero oxygen. Well, as you get near to the cabinets, as it turns out, there's quite a bit of leakage. And of course, the researchers are constantly going in and out and opening doors, and the nitrogen is spreading. If I hold one of our monitors up next to the orange case, you can see that I'm already down to 14.5% oxygen. So that's not enough to knock you down. It is enough to produce symptoms. If you're working in that area, you're going to be pretty tired. You're way below the oxygen deficiency threshold, which is 19.5%. So that's a pretty low concentration. That's a dangerously low concentration of oxygen. But it could quickly get a lot worse. If you have a breakdown in the ventilation in this little room or a power outage, you're going to get down below 10% pretty quickly. And when you're down below 10%, you're going to lose consciousness pretty quickly. By the time you're down at 6%, you take one breath and you're going to lose consciousness instantly. So types of laboratories, analytical, quality, classrooms. Don't forget university and college classrooms, clean rooms. Clean rooms often need special attention regarding the containment of the air in the clean room, the air quality, and also particulates, as well as everything else that's going on. Clinical and medical labs, pathology, serology, histology, virology, sounds like a song. Bacteriology, molecular biology, PCR, that's polymerase chain reaction technology labs, microbiological cell and tissue culture with controlled temperature, humidity, oxygen, and carbon dioxide level. Research and development labs where who the heck knows what's going on. It could be almost anything. Types of atmospheric hazards, extremely wide range. But number one on the hit parade is going to be oxygen deficiency due to displacement, followed by combustible gas and VOCs, and especially alcohols, solvents, and other sanitizing chemicals like ozone, chlorine dioxide, ethylene oxide, or chlorine. Boy, that's easy for me to say. Don't overlook, especially in clean room and areas where you are especially sensitive with regards to biological contamination. A lot of the time you have workers that will be working in supplied air respirators, which require a grade D breathing air source. So if you have people on supplied air respirators, do make sure that you are supplying them with grade D breathing air, and if necessary, that you have a portable or wall-mounted purification panel, and that you're monitoring that airstream for carbon monoxide and or dew point and or often oxygen as well. As I mentioned, the solution can be either fixed or portable, or a combination of fixed and portable industry. So when the hazards are generally present everywhere all the time, like at a petrochemical plant, typically you'll have every worker in that area on the chemical plant side of the refinery equipped with either minimally a single channel H2S monitor or a 4-gas monitor that's looking for H2S, combustible gas, oxygen, and carbon monoxide. When the hazards are present in specific areas, either potentially or on a regular basis, then you should definitely think about fixed gas detection. For instance, in our earlier example, if you've got a room with 3 or 4 industrial sized dewars full of liquid nitrogen, then you best have a fixed monitoring system. You wouldn't really want to deal with that hazard only with the issuance and use of personal oxygen meters. So often the best solution includes both fixed and portable. It all just depends on what's going on in the area. Again, your choice of combustible sensor is really, really important, and which type of LEL sensor is best really does depend on what's going on. Traditional catalytic or Pallister LEL sensors, the reason they call them Pallister type sensors is the active bead. It looks like a little pellet that's made out of that alumina material that is then treated with the catalyst that allows the oxidation to occur. It detects gas by burning it basically in a controlled catalytic way on and in that bead, which heats that bead up to a higher temperature than it started out. So the concentration of gas depends on, or how you register the concentration of gas is by how much that bead is heated up beyond the temperature that it started out at. So traditional catalytic LEL sensors, as we discussed, the alumina material in the active bead makes them particularly prone in pharmaceutical applications to being burned out by exposure to methanol. Methanol is not good for traditional catalytic LEL sensors. Also, traditional LEL sensors require the presence of oxygen in order to detect gas. Some substances like silicones, we're not talking about fully cured materials that are made from silicone. What we're talking about is stuff like Armorol or RTV or silicone lubricants or other materials that release the silicone as they are curing. Um, your options besides the catalytic LEL sensors are IR LEL sensors. They come in full-size version with a long optical path. These kind of sensors require a little bit more power, but they are better for dealing with olefins and unsaturated and aromatic hydrocarbons and VOCs. IR LEL sensors can never ever be used to measure hydrogen, and the ones in portable instruments are not able to detect acetylene either. So if you have either of those two gases, then you need to make sure that one of the other sensors in the instrument is going to be capable of picking them up. So the full-size IR LEL sensors are really good at measuring unsaturated as well as saturated type hydrocarbons and alcohols. The miniaturized low power sensors, IR-LEL sensors that they put on a chip, these MEMS sensors, the benefit is that they take very, very little power to operate. The bad news is they are primarily designed for the detection of methane, and they are often not very accurate for other types of combustible gases. Another type of sensor that we're finding in portable gas detectors is the MPS sensor. That stands for Molecular Properties Spectrometer. And these detect gas by looking at the difference of the density of the gas in the affected area versus the density of fresh air. So lighter than air combustible gases like methane will have lower density, and you can tell the difference. Heavier than air combustible gases like propane and pentane and alcohols are heavier than air. So again, you have a detectable difference in the density compared to fresh air, but you do have to be careful with MPS type sensors in confined spaces where the gas of interest is not being mixed into fresh air with 20.9% oxygen and balance nitrogen. It turns out, for instance, that propane has the same molecular weight as carbon dioxide. It's about 44 grams per mole. And 21,000 parts per million of propane looks like 100% LEL. 21,000 parts per million of carbon dioxide also to the MPS sensor looks like 100% combustible propane gas. So you do need to think through how you are going to use these sensors. They're great for some applications, especially when you're mixing the gas of interest into fresh air, but not recommended for use in confined spaces, and especially not recommended for use in inertion procedures. Again, the advantages and limitations of the catalytic type LEL sensor: predictable, well understood technology, been around for about 75 years, predictable cross sensitivities. It's able to detect hydrogen, acetylene, and unsaturated hydrocarbons in general, all those olefins and aromatics of interest. Disadvantages: the catalytic sensor uses more power than the other sensors. It has a lower and slower response to larger molecules, including alcohols. It can take up to 5 minutes for the catalytic sensor to reach its final completely stable 100% T100 reading. It can take 5 minutes to get there for something like ethanol. That's a pretty slow rise. And it's easily poisoned by silicones, by methanol. It's can be burned out by exposure to alcohols, and it needs at least 10% oxygen in order to detect gases correctly. For IR-LEL, again, you have advantages and disadvantages. Here you can see a full-sized IR-LEL sensor. Here you can see a miniaturized MEMS chip-mounted type sensor on the inside of this housing. You have a tiny, teeny little chip, which is the actual sensor. It's very small, and the optical path length is less than a tenth of a millimeter. The optical path length in the full-sized sensor is actually about 7.5 centimeters. So you have a lot more opportunity for absorbance to occur. But when it comes to IR-LEL sensors, they can't be poisoned. They don't require oxygen. They can be used for high-range measurement, and they respond very well to very large molecules that cannot be measured easily by means of a standard sensor. The disadvantages: the molecule has to include chemical bonds that absorb at the wavelengths used for the measurement. If you don't have absorption of infrared light at those wavelengths, then the gases are invisible. So not all combustible gases can be detected, and you can't detect hydrogen or acetylene ever. When we're talking about the IR-LEL sensors, especially the miniaturized IR-LEL sensors, make sure that you actually read the owner's manual. The owner's manual is something that is, when you go to the certification agency like UL or CSA, they are going to insist that you add certain warnings, and they're not going to give you the certification until they finally sign off on the owner's manual. That's part of the audited material. So generally speaking, you can trust. You may not always be able to trust what my friends and colleagues among the professional salesman community, I wish you could, but just in case, have a look at the owner's manual. The owner's manual you can trust. This is the page of warnings for the IR-LEL sensor in a very popular compact miniaturized 4-gas instrument that uses an IR-LEL sensor. So if you are talking about methane and you expose it to 20% LEL, your reading is on the money. For propane, 20% exposure can get you a reading of anywhere between 15 and 45. It's not very predictable. For pentane, it can be anywhere again between 15 and 45. For hexane, it can be anywhere between 8 and 28. That's pretty vague for making an important decision as to whether or not you are at or below 10% LEL, where most of our instrument users take action. For methanol and ethanol, the sensor responds very well, but again, it's kind of a qualitative response. You can have a response anywhere between 6 and 26. For hydrogen and acetylene, no response whatsoever. Different IR-LEL sensors are going to have different advice. The full-sized sensors have a much more predictable response to the gases that absorb less strongly, and also they have more sophisticated, they're more expensive, and they have more sophisticated lookup tables for moving from one gas to another gas. VOCs figure prominently at chemical plants and especially in laboratories and pharmaceutical plants. So what is a VOC? It's a wide category that includes everything from jet fuel and diesel to specific chemicals like hexane and toluene, butadiene, xylenes, and benzene. So if you have these VOCs on site, then instead of a 4-gas instrument, probably you should consider adding sensor number 5, which is going to be a PID sensor that will allow you to take action and sound the alarm when you hit the toxic exposure limit, rather than waiting to hit the percent LEL, lower explosive limit concentration alarm. And if you look at ethanol, ethanol is a good example. The OSHA-PEL, the NIOSH-REL, and the TLV are all 1,000 parts per million. If you set the LEL alarm for ethanol to go off not at 10% LEL, but at 5% LEL, that would be 1,650 ppm. Well, that's going to be higher than the TWA limit. If you set it to go off at 20% LEL, then that's over 300% higher than the toxic exposure limit. That's too high. So for all of these VOCs, every dang one of them, where you have a published exposure limit, you're going to exceed the toxic exposure limit before you get to the 10% LEL or even 5% LEL. So this is, you've got a lot of thinking to do if you're considering changing your gas detectors or expanding your gas detection program. This is really the golden age of gas detection. There have been more changes in the last 5 years, I think, than in the prior 40 years. There are new products, new sensors, wireless communication is reaching maturity. You have integrated fixed and portable networks where you don't have two separate networks, you have a blended network that includes both. You have third-party support through call centers and coordinated emergency response, third-party help with record keeping and notification, and internet-based maintenance programs. So these are all relatively new things to consider, new developments. So you've got some research to do to make sure that where you're going with your instrument needs really is where you want to be going. Before making a change, you really should understand how your current products are working. A lot of times, managers at large facilities are, you got a lot of things on your plate. You may not know every little detail about how the current instruments that are in service are doing. So your job needs to be to include finding out. If you are not sure, make sure to find out the brands and models in current service. You might be surprised. Make sure you understand the current capabilities, the strong points, the weak points of the products that you're currently using. Ask the manufacturers or distributors of the products that you work with or that you are interested in for help. Download specification and comparison charts if the manufacturer has them. Discuss the ways that the manufacturer and distributor can help you, and it's their job to help you. You have every right to demand a high level of help and support. Number one critical starting point question to ask yourself is, are you happy? Are you experiencing a lot of problems? How old is your current equipment? What are the features you may have heard about that you are interested in? What brands and models of gas detection are you considering? And what are the alternatives? Do you know what the alternatives might be? Distributors are a great source for product information. When in doubt, or with regards to advanced technical questions, I would urge you to go right to the manufacturer. Manufacturers have absolutely first-rate, really excellent product managers and specialists that can take the technical information to the next level. Avoid being overly focused on price, at least to begin with. Eventually, the decision of whether to proceed involves price and affordability. However, there is a difference between the initial purchase price and the true cost of ownership. So make sure that you understand what the benefits of the new equipment are, how they will affect the overall cost of ownership of the equipment, and that may change your thinking about the initial purchase price. It may be better to spend a little more upfront and have a much more affordable uh, uh, piece of equipment over the 6 or 8 or 10-year life of the instrument. Make sure you identify the cost of ownership issues. Are you spending a fortune keeping your current equipment in service? Are you being charged an exorbitant monthly fee? Do you trust your gas detectors? Do you have a lot of sensor failures? Do you have battery problems? How often are you testing and calibrating your instruments? It is really important. OSHA agrees, the test laboratories agree, the manufacturers agree. You should perform a bump test on the basic sensors at the very least in your instruments before each day's use. So it's going to be hard to do if you're sending instruments in once a year or twice a year to a third-party calibration service. If you agree that you need to perform a bump test before each day's use, do you have the gas that you need and the equipment that you need together with the instruments on site where you can actually perform the bump test? So once you get into all of these issues and you really think about whether you do in fact want to proceed with bringing in those new instruments, now you can bring price into the equation. Uh, don't be afraid of fixed detection systems. Fixed detection systems are not magically hard to specify. They're not magically hard or difficult to maintain in service. In fact, once they're up and running, they tend to be service and problem free. A lot of what I would encourage you to do with regards to fixed depends on starting from a point where you actually know your needs. All of the manufacturers have a simple to fill out form, a fixed system questionnaire that will just specify a few important things that you might not have thought about when you set out thinking about whether you needed a fixed system. Do you want to wire it by integrated by digital means, by Modbus, by HART? Do you want to connect the gas detectors with the controllers by means of a 4 to 20 milliamp loop connection? Do you even need a controller, or do you already own a PLC? If some of these terms you don't know what that means and you've never heard those terms before, don't despair. That's why you have friends at the manufacturer and friends at your integrators and distributors to work with who do have the vocabulary and who can help you through the process of filling out that questionnaire. Make sure you have a look at how you're handling confined space programs. Again, the era of limiting your confined space monitor to the basic 4 gases, well, that was then. Nowadays, a lot of instruments, especially at chem plants, at pharma plants, at laboratories, include a 5th, a 6th, or even a 7th channel or sensor of gas measurement. It's all available for you. You just need to verify what it is that you need to measure. Uh, Watch out for compatibility issues. There are some combinations of sensors that you don't want to have in the same multi-sensor instrument. For instance, nitrogen dioxide and sulfur dioxide. Exposing an SO2 sensor to nitrogen dioxide drives the reading really deeply negative. You'll have a downscale negative alarm. And the opposite is true as well. Each sensor drives the other one, each gas drives the other respective sensor deeply negative. So a lot of times it just makes more sense to keep the odd gas out in a separate instrument. If you have an instrument or a sensor that just takes a little more work, harder to calibrate, more expensive to calibrate, more expensive to do a bump test on, then keeping it in a separate instrument which you use only when you need to use it makes a lot of good sense. Again, watch out for alcohols in big pharma. I really urge you to take a look at using an infrared sensor if you are needing to measure methanol in particular or other alcohols. Make sure that you address how you're going to use the instrument for confined space measurement where you need to have a pump. You can have a diffusion instrument. You can have an attachable pump that draws power from its own battery pack. You can have an internal pump that draws power from the instrument battery. And in this case, it has a slide switch that when you have the pump off, it operates in diffusion mode. When you have the pump on, you're pulling a sample through the hose and probe assembly. So you have a few different ways to go. Batteries are a big issue. Lithium-ion batteries, unfortunately, are frequently in the news. They are filled with a flammable electrolyte, and if you heat that up to a high enough temperature, it will spontaneously catch flame, catch fire, and you've got a lithium fire on your hands that is really, really hard to put out. You have a couple of alternatives. You have alkaline batteries, you have nickel metal hydride batteries that are not filled with a combustible electrolyte. So make sure that you've thought about it. Again, when it comes to bump tests, make sure that if you are committed to performing a bump test before each day's use, you have the materials living conveniently in the same place where the instruments live. Make sure if you're using a docking station that the docking stations are located conveniently where people can actually use them to perform the tests. If you are interested in wireless, there are several different methods of wireless communication. Some of them are good for short range, but require a collecting device like an intrinsically safe tablet or an intrinsically safe cell phone that can then transmit the information to a further off location. The alternative you have is a true RF wireless transmitter that typically has a much longer transmission distance and can go directly back to a base station or a truck or a computer, or in this case, a intrinsically safe server. that can be in the hazardous location or with the attendant or confined space supervisor at the point of entry. So you've got several options, and you have a lot of different options with regards to what you can do with the information when it's acquired. So a lot of things to think about. You have a right to good after the sale support. Satisfaction is a function of ongoing support. So if you are happy to buy products from the internet and don't expect to ever see a human being on site to give you a hand, God bless. That's an available option. If you need the after the sale support, then remember that's the job of distributors and manufacturers to make sure that you have it. So part of the evaluation process should be, how are they able? Are they willing? Are you going to get the after the sale support that you deserve, need, and want? So that comes to the end of the webinar. Thank you very much. Drop me a note, give me a call, give anybody at GFG a note or a call. We are passionate about gas detection. We are here to give you a hand. And Samantha, thank you very much. It's back over to you.
Well, thanks, Bob. That was a lot of information to take in. If you wanted, we could take a few questions before we wrap it up for the day.
Fine with me if it's okay with you.
Sounds good. So this is a great question, and it's one I've always wanted to know. How long does it actually take to perform a bump test?
Well, if you're using a docking station, so here typically we are putting the instrument into the docking station, and the docking station does its thing. And its thing can be to perform a daily test, or if it's now time for a full calibration, it can automatically do the full calibration. But if you pick the daily test, technically what the docking station is doing is going a little bit beyond a bump test. A bump test, according to OSHA and the ISEA, the International Safety Equipment Association, a bump test is purely qualitative. All you're doing is exposing the sensors to gas in a concentration of gas that is high enough to cause the activation of the alarms. So you're not looking for the accuracy of the reading, you're verifying that on that particular day, the sensors respond to gas. So with the docking station, the evaluation includes the accuracy of the reading as well as testing the proper activation of the alarms and the flashing lights and the vibrator and anything else you have built into the instrument. And typically that's going to take for a 4-gas instrument probably 20 to 30 seconds. If you've got a PID in there, then it might take 45 to 60 seconds to get through the daily bump test. But if you are doing a manual qualitative bump test, probably the easiest way to see how long it lasts would be to show you. So here let's try this at home to do the movements in a mirror version. It's harder than you would think. But anyway, what you're doing for a manual bump test is you're taking the adapter. Typically, we're talking about a 4-gas instrument, putting the adapter onto the instrument. And now all it takes is about a 2-second burst of gas. And all I want to do is see all 4 alarms go off. So I have given it 2 seconds worth of gas flow. And as you may or may not easily be able to see, all of the sensors have momentarily gone into alarm. So about 2 seconds worth of gas and about 10 seconds to perform the bump test, and OSHA is happy. Make sure that you record the results. If you're doing a manual test, then you probably want to have a log. If you're using a docking station to do the daily test, then the docking station will record and retain the results. But good question.
That was excellent. I love when you pull out the gadgets, Bob. It's a really good visual representation, and I think it really helps people understand how to use the application, what the process should look like or does look like, and then where to spot those errors if there are any.
Yes, ma'am.
Awesome. So take another couple of questions. Why is alcohol so hard to detect with a standard LEL sensor?
Well, for a couple of reasons. One is the standard LEL sensor is protected by filters and flame arrestors, and there are barriers for the alcohol that the alcohol has to diffuse through before it can get into the sensor and finally get oxidized on the active bead. So it just takes time to get there. It takes even more time if you have a standard catalytic sensor with a protective filter that's designed to remove silicone, which is one of the very worst of the combustible sensor poisons. So it really can take 5 or 6 or 7 minutes for the even ethanol to get into the sensor to respond, and for your readings to stabilize. And that takes a long time. That's kind of too long. And then the other issue is that alcohol is very hard on the active bead in the sensor, especially methanol. Methanol and alcohol, if you're interested, let's see, this might be a kids don't do this at home kind of deal, but if you happen to have shredded aluminum and you pour some high test methanol on it, um, it's really going to show some corrosion damage pretty quickly. Anybody that uses methanol fuel in a blower in your yard, you'll notice that all of the stuff that's made out of aluminum in the blower, it does not like being exposed to the methanol. It wears out pretty quickly. So you're going to burn out sensors with methanol. With infrared, you don't have all of the filters that you have to go through. There's nothing that you can poison or burn out, and the sensor will respond very strongly to the alcohol and accurately to the alcohol if you've got the right sensor. The only thing that you always have to remember, if you have hydrogen, then you should consider adding a direct reading substance specific electrochemical hydrogen sensor to that multi-sensor instrument along with the IR LEL sensor. By the way, in pharmaceutical and laboratory applications, there's a lot of carbon dioxide, and you can have a dual channel CO2 and IR LEL sensor. So you get two readings, two discrete signals for the price of one sensor, and you still have room for a PID. So one of my favorite configurations, in fact, for a pharma instrument would be O2, hydrogen, PID, carbon monoxide and hydrogen sulfide, and IR, LEL, and CO2. And that's actually easy to do. The days that you were limited to 4 channels only that was then. We can cram a lot of detection technology into a single instrument nowadays.
That's awesome. I know we all love a good 2-for-1 deal, so good to know. So we're going to take one last question. We're going to wrap it up for the day. Can I
Awesome.
switch sensors in my instruments depending on what I need to measure?
Yes, you can, but when you, let's say that you take my advice and you decided not to put the SO2 and NO2 sensor in the same instrument, even though you could. So I have NO2 today and I want to switch it over to SO2. Well, to do that, it's pretty easy. You take the housing apart, you access the sensor, you unplug one, you plug in the other, but you have to go through a warm up and calibration process before you can actually use the sensor. Even if you're keeping sensors in an external so-called warm-up box, disconnecting the sensor even for a moment or two while you're pulling it out of the box and then putting it into the instrument allows that sensor to destabilize quite a bit. So by the time that you let it settle back in for 30, 40 minutes, and then you really do need to put it through a full calibration. By the time you do all that, um, that's a lot of effort just to reconfigure your instrument. If you're at a petrochemical plant where you've got a shutdown coming up, we've got large customers with 50 or 100 instruments, and they will set them up with a certain slate of sensors for a certain mission. And when they have a different shutdown, maybe they will use a different slate of sensors. So that's a great time to change the sensors. But generally, you're probably going to be better off putting the most commonly used sensor into the multi-sensor instrument and keeping the less commonly used sensors in a separate single sensor instrument, or have a multi-sensor instrument with a different configuration of sensors. I think that's
Perfect.
about it.
I love it. Well, I want to give a huge shout out to AD Safety Network and GFG for allowing us to have this very great presentation as always, and I want to thank you, Bob, for being here today. Again, if you're looking for the opportunity to connect with peers and industry trendsetters through our global EHS audience, be sure to attend our 3-day virtual safety conference and expo, October 19th to 21st. This free event brings together EHS professionals in a platform that provides a place to learn, collaborate, connect, and have fun. Last year we had over 4,000 attendees, 30 exhibitors, 30 speakers. Don't miss out on registering for this year's Safety Connect. And don't forget you'll receive 1.7 CEUs for attending. Register at industryconnectsafety.com. On behalf of Safeopedia, I want to thank everyone for being here today, and I want to thank all of the dedicated safety professionals for the great work they do on a daily basis. As always, stay safe, take care, and we'll see you next time. Bye, everyone.
Bye bye, guys. Thank you.